Hello, my name is Charles Mulligan from the Department of Pathology at St Jude Children's Research Hospital. I thank the organisers for the invitation to present at this symposium, and I'm pleased to give this talk entitled The Many Genomic Roads to Acute Lymphoblastic Leukemia. Acute lymphoblastic leukemia is the commonest childhood tumour. Most children are now cured of the disease, but relapse does occur in a substantial minority of individuals, and when it occurs, it's very difficult to induce back into a remission and long-term disease-free survival. And for that reason, childhood ALL is the leading cause of cancer-related death in the young. Furthermore, many of the drugs that are used to treat ALL have been in use for many years now, if not decades, and we have a relative dearth of new agents targeted to specific genetic alterations or vulnerabilities in different subtypes of ALL. So clearly there is a need for better therapeutic approaches. What I would like to do today is to give some history about what's been previously understood about the genetic classification of leukemia, and then give some examples from a nearly overwhelming amount of work that's occurred in the last few years using integrated genome and transcriptome sequencing that has revolutionized our understanding of the genomic landscape of ALL, both in children and adults, and has led to a number of new diagnostic and therapeutic approaches. So until some years ago, our classification of ALL was really driven by conventional cytogenetics, that is karyotyping, which identified a number of alterations including hyperdiploidy and hypodiploidy, as well as a small number of recurring chromosomal rearrangements that define specific subtypes of ALL in children and to a lesser extent in adults. What was clear from this knowledge is that there was a substantial amount of individuals across the age spectrum that were not accurately identified using genetic approaches. This is particularly true in adults. Moreover, it was clear that these cytogenetic aberrations shown on the left were very strongly associated with very distinct transcriptional signatures in leukemic cells. And an example of this is shown on the right using microarray gene expression profiling. What has now changed has been the advent of genome sequencing, and particularly transcriptome sequencing, which has really transformed our understanding of the genetic drivers of ALL. One of the first of these studies was a collaborative one published between the St Jude WashU Pediatric Cancer Genome Project and the Children's Oncology Group Target Initiative that identified PH-like acute lymphoblastic leukemia as a very common form of genetically heterogeneous form of ALL, particularly common in older individuals. And since that time, over the last few years, a number of studies in children and adults of various size have reported a number of new targets of genetic rearrangement in ALL, including transcription factors, kinases, and other genes, several of which appear to classify new subtypes of ALL. What I show on this slide is a transcriptome-based report of classifying ALL using almost 2,000 transcriptomes from approximately 2/3 in children and 1/3 in adults, every case subjected to transcriptome sequencing and a proportion to SNP array profiling and whole genome sequencing to fully understand the nature of genetic alterations. This was a collaboration between multiple cooperative groups, the COG and St Jude, as well as a number of adult cooperative groups. And a goal here was to see how far transcriptome sequencing could be utilized to classify the landscape of ALL, and also to determine whether it might be useful as a diagnostic approach. And I'll come back to that thought at the end of the talk. What one is seeing here is a t-SNE plot, often used to show single cell gene expression profiling data, but each data point here is a case of ALL, tumor transcriptome sequencing, color-coded according to subtype. Many of the previously identified subtypes are here visible, rearrangements of MLL, TCF3 PBX1, hypodiploidy, ETV6 RUNX1, and so forth. And we can immediately see a number of other observations, for instance, new subtypes of ALL driven by rearrangements of transcription factor genes, for example, MEF2D, DUX4, and ZNF384 that define distinct subtypes of ALL. A number of other subgroups that were identifiable also with distinct gene expression profiles, perhaps not as common, but with so many more cases, we could now start to see these additional subtypes. Cases in children with rearrangements of NUTM1 associated with favourable outcome, cases in adults with rearrangements of BCL2, MYC and BCL6, rearrangements also seen in a subset of diffuse large cell lymphoma, although here describing a group of pre-B cell ALL. And additional subtypes of leukemia driven by transcription factor genetic alterations. I'll come back to some of these themes later in the talk. There are two groups here defined by alterations of the B cell transcription factor gene PAX5, one group with a single point mutation P80R, and one group with more diverse alterations called PAX5ALT with a number of different mutations and rearrangements. We are seeing this theme of single point mutations describing subtypes of leukemia more frequently, another example of which is shown here for the Ikaros or IKZF1 gene. And then we have the quite large and genetically diverse group of kinase-driven leukemia defined by either the BCR-ABL fusion from the Philadelphia chromosome or these PH-like cases that I'll come back to in a moment. So in the next few slides, I show some examples of these data and make some points as to why the lesions are important diagnostically, biologically, and therapeutically, and also to make the point that really we need sequencing-based approaches to accurately identify these patients with these new alterations. And this is that many of these alterations are really not amenable to conventional cytogenetic analysis because the alterations may be cryptic or subtypes are polygenic with multiple co-lesions or multiple phenocopying lesions defining the same type of leukemia, or as I've mentioned already, there can be mutational drivers of leukemia subtypes. So some examples. This is a nice example of a common theme here. This is a subtype defined by rearrangements of this gene, MEF2D, myocyte enhancer factor 2D, that's fused to a number of different genes, most commonly BCL9, but a number of other partner genes as well. It has a distinct immunophenotype, as shown here, with negativity for CD10 and positivity for CD38. And we can see, pursuing some of the biology of this form of leukemia, some interesting observations that are relevant to leukemogenesis and therapy. So, for example, MEF2D regulates a program of gene expression in muscle development with overexpression of a gene called HDAC9 or histone deacetylase 9, which is what's seen in this subtype of leukemia with reciprocal downregulation of another MEF gene, MEF2C. We observe that these fusions are transforming in reporter assays and colony replating assays. And of therapeutic interest, we see that perhaps because of this elevated expression of HDAC9, that human leukemia cells with this MEF2D rearrangement are exquisitely sensitive to histone deacetylase inhibitors, such as panobinostat, as shown down here. So an example of where this is not really a direct mutation or fusion-directed precision medicine approach, but a vulnerability that's arising from the deregulated pattern of gene expression induced by expression of the fusion. An important point of diagnostic relevance, again, is this notion of what we call phenocopies, where we see non-canonical fusion oncoproteins or other genetic lesions that are simulating the founding lesion, and we see this right across the landscape of ALL. A striking example of this is ETV6 RUNX1 leukemia, where we can see color-coded here a number of cases that are color-coded grey or alternate colors because they are clustering with ETV6 RUNX1 leukemia and is shown in yellow, but they don't have that fusion. This is a phenomenon that's seen broadly to greater or lesser degree, with most subtypes having a proportion of cases that are simulating the canonical subtype of leukemia. The most striking of which is PH-like leukemia that outnumbers PH-positive leukemia, at least in children, by several fold, but we see this less frequently in the other subtypes of leukemia. Coming back to ETV6 RUNX1 leukemia, if we look at the lesions that are driving this form of leukemia, they make perfect sense. We see a number of different rearrangements targeting ETV6 itself, or a number of other fusions that are targeting other genes, including other ETS family transcription factor genes like ETV6. These cases thus have similar biology, similar clinical behavior, and it's important to diagnose these patients for risk stratification at the time of diagnosis. A new finding is one that I've mentioned before, which was really not appreciated in ALL, was that it's not just gross chromosomal alterations, but point mutations that can be the first initiating events in leukemogenesis. Two examples of this are the PAX5P80R missense mutation and the Ikaros N159Y mutation. So if we look in a little more detail at this observation, this is not a new phenomenon that PAX5 would be altered in leukemia. Indeed, we had seen many years ago that this B-cell transcription factor gene that's required for B-cell development is broadly mutated across the spectrum of BALL, most commonly by deletions of part of the gene or the entire gene, as shown by these bars here, or point mutations affecting the DNA binding domain or the transcriptional activation domain, as shown here, or a number of fusions which are less common, but now with many more studies we've seen over 20 different partners of rearrangement of PAX5. Now, with the exception of these PAX5 fusions, we had inferred that these point mutations or deletions were secondary events, important in arresting B-cell development, important in driving leukemogenesis, but not the tumor-initiating events. And this was based on the observation that many of these point mutations and deletions were seen in the context of other leukemia-initiating drivers, for example, ETV6-RUNX1. However, we now see this crisply transcriptionally defined group of leukemia, all of which cases have this missense mutation P80R. And this plot here shows the protein domains of PAX5, the DNA binding domain, the transcriptional activation domain is shown here, and the mutations in PAX5 that are occurring in this P80R defined group. Several cases also have second hit mutations, most of which are loss of function due to a frame shift. But if we contrast this with the remaining landscape of BALL, we see that there are many other mutations, including in the DNA binding domain, many of which are quite deleterious for PAX5 DNA binding and function, but they're not seen in this group, P80R. So there is something distinct about this subtype of leukemia that's defined by the single point mutation. If we look at the genetic co-lesions in this form of leukemia, there are a couple of important observations. All of the cases have the P80R mutation as well as a second alteration of PAX5, either a loss of function frame shift mutation or a deletion of the wild type copy of PAX5, most commonly by either a broad or focal deletion of the PAX5 locus itself. There is quite a high frequency of signaling pathway activation, particularly the RAS pathway, but also activating mutations in the JAK-STAT signaling pathway as well. So these observations of a universal occurrence of a PAX5 mutation, the second lesions that appear to delete the wild type copy of PAX5, prompted us to formally address the question as to whether these mutations are indeed leukemia initiating. And we addressed this by CRISPR-Cas9 genome editing, where we used this technique to introduce several mutations into the mouse germline and then observe mice for the development of leukemia. The results here were quite striking, particularly because leukemia is typically very difficult to model in the mouse, at least B-cell leukemia is difficult to model in the mouse. And here we see, without introducing any other co-lesions, we see that mice heterozygous for this PAX5P80R mutation developed leukemia within 6 months, and mice that were homozygous developed leukemia in approximately half that time. This is in contrast to a second mutation called G183S that's found in familial BALL that I'll come back a little later in the talk, but here we found that this presumably is a very mild mutation and it did not generate leukemia, at least not without secondary mutagenesis. The leukemias were pre-B-cell in phenotype, as we see in human leukemia, and when we performed comparative genomics of the mouse tumors, we found that there was a broad or focal deletion of PAX5, or here in a case of a mouse with homozygous P80R, there was indeed amplification of the mutated copy of P80R, increasing its oncogenicity. So these findings fit very well with what we see with human leukemia, that this is a founding mutation. There is a stepwise road to leukemogenesis with acquisition of these second hits on the road to leukemia, including second hit alterations of PAX5, thus proving the point that this is indeed a classifying and leukemia initiating a mutation in VALL. I don't have time to go through all of the subtypes, each of which is interesting in their own right for various reasons, but now if we come back to look at a landscape of BALL, we can refine the classification much better. We've moved from less than 10 subtypes to now over 20 subtypes of leukemia. Even this slide is a little out of date now, as we've identified a couple of uncommon additional subtypes of leukemia, but this represents the main subtypes of leukemia that are now recognized. It's important to stress the point that these subtypes are highly age dependent. We see a predominance of good risk subtypes of leukemia in children, such as hyperdiploidy and ETV6 RUNX1, as well as some other subtypes of leukemia, ETV6 RUNX1-like leukemia, as mentioned before. And with increasing age, we see a rise in the frequency of PH-like leukemia, and with a further increase in age, a continual elevation in the rate of BCR-ABL positive leukemia, such that kinase-driven leukemias, be they pH positive or pH-like, account for the majority of cases of adult leukemia, and good risk subtypes of leukemia seen in children are very uncommon. Additional striking alterations seen in adults is that MLL leukemia is quite common, as is low hyperdiploid leukemia, an observation that was not really identified until quite recently. These subtypes are strongly associated with outcome, both in children and adults, and indeed many of the new subtypes further refine the risk classification and prognosis of ALL. For example, two new subtypes, DUX4 rearranged ALL, the PADR group, are associated with relatively favourable prognosis. This is also seen in adults, where quite strikingly DUX4 rearrangement is associated with very favourable outcome, with survival exceeding 80%. In contrast, we now have a handful of subtypes that are associated with the majority of relapses in adult leukemia, pH positive, MLL rearrangement, pH-like, hyperdiploid leukemia, and this uncommon but very important subtype of leukemia that nearly universally shows induction failure with dual alteration of BCL2 and MYC. Perhaps the greatest outstanding problem in BCR-ABL positive, the greatest outstanding problem in B lineage leukemia is pH-like leukemia. A very large number of studies now have consistently shown that, firstly, this form of leukemia is very common. It rises to over 20% in adolescence and remains at that prevalence right across the age spectrum. It's universally associated with high-risk features such as high rates of minimal residual disease or poor outcome. And it's also exceedingly genetically diverse, and this diversity continues to grow, where there are now over 16 different cytokine receptors and tyrosine kinases that are subject to mutation, commonly rearrangement, but also point mutation and deletion, involving a large number of partners of the rearrangements, over 60 and growing, as shown here, with the greatest diversity seen for ABL itself, JAK2, and PDGF receptor beta. Now, despite this diversity, one can distill down this heterogeneity into several pathways that activate key signaling outputs. So the most common subtype of pH-like ALL are those cases that are driven by activating lesions involving the JAK-STAT signaling pathway, the most common of which is CRLF2 rearranged leukemia, where this protein is enhancer hijacked. It's expressed in the B cell lineage. It forms a dimer with interleukin-7 receptor alpha chain, and these cases, about 50%, have concomitant activating mutations of the Janus kinases, particularly JAK1 and JAK2. However, there are additional alterations that drive JAK-STAT signaling. There are mutations of the IL-7 receptor itself. There are very distinctive and interesting truncating rearrangements of the erythropoietin receptor that deregulate its expression and result in constitutive activation and loss of receptor downregulation because of the truncation, as well as some other lesions, for example, conventional chimeric fusions involving the Janus kinase family. The second class of pH-like leukemia are those with ABL class alterations, that is conventional chimeric rearrangements involving ABL1, ABL2, CSF1 receptor, or PDGF receptor alpha or beta. And then there are a number of less common but very important alterations that target genes that are, some of which are very well established in leukemia, such as FLT3. We see both conventional ITD mutations, kinase domain mutations, as well as chimeric rearrangements of FLT3 we're now seeing for the first time. ETV6 and TRAC3, which is a rearrangement seen in many tumours and other childhood tumours as well. And less well studied genes in leukemia biology in terms of targets of rearrangement, such as B-cell linker, a component of the B-cell receptor signaling complex, and PTK2B, both of which are targeted by rearrangements. It should be emphasized that there's a large body of preclinical literature showing that these lesions do indeed activate the predicted downstream signaling pathway. They also activate additional pathways such as PI3K, BCL2, etc., that represent additional potential targets for therapeutic intervention. And these strategies are now slowly moving towards the clinic. There is a diversity in response to therapy in PH-like leukemia. So I show a couple of examples of this. At one extreme, we have ETV6 NTRK3 leukemia, which is seen in 1 to 2% of leukemia cases where there's this chimeric rearrangement of ETV6 to NTRK3. The cells are cells expressing ETV6 NTRK3 are exquisitely sensitive to larotrectinib, a NTRK3 inhibitor, or here a plexicon agent that also inhibits CSF1 receptor, which is targeted by fusions in ABL class PH-like leukemia. From mouse models that I'm not showing here, if we knock in ETV6 NTRK3 in the B-cell lineage, we show striking development at a very early age of B-cell leukemia. And both engineered mouse models as well as human leukemia xenografts are exquisitely sensitive to larotrectinib, and these data are shown here, where these are untreated animals showing leukemia burden, and these are animals treated with larotrectinib, where we get essentially complete suppression of leukemia cell growth. If we stop treatment, the disease will come back, but then it will completely respond again. And indeed, if we start therapy initially for sufficient duration, we can completely eradicate the disease with no recurrence after stopping larotrectinib therapy. So this is a good and striking example where we have a monogenic driver of disease and a complete response to therapy with single agent tyrosine kinase inhibition. Contrast to this is CRLF2 deregulation, which I mentioned is the commonest form of JAK-STAT-driven PH-like leukemia. It's also the commonest form of PH-like ALL overall. And I've mentioned some of this information already. CRLF2 is in hearts are hijacked. It forms a receptor with IL-7 receptor alpha. It's commonly seen in both PH-like as well as Down syndrome-associated leukemia. And it's driven by these two main enhancer hijacking events, of focal deletion adjacent to the gene that brings in opposition to this enhancer of this P2RY8 gene, or an IGH enhancer rearrangement that also drives expression of CRLF2, resulting in this activation of downstream signaling pathways, not just the JAK-STAT pathway, but also the ERK and PI3K pathways as well. And this is a form of leukemia where more work is needed. I show a couple of examples of data here for JAK-STAT-driven leukemias where more conventional chimeric fusions, two examples of which are shown here, PAX5-JAK2 and another fusion of JAK2, show some response with TKI monotherapy, but good response with combinatorial therapy with a backbone of conventional chemotherapy, such as with dexamethasone, as shown here and here. In contrast, TKI therapy for CRLF2 rearranged leukemia is much less and more variably efficacious, with some tumours in preclinical models showing a partial response and some showing no response. So a number of different TKIs are being tested in both adult and pediatric PH-like ALL, particularly dasatinib for ABL class fusions and ruxolitinib for JAK-STAT-driven leukemia. There are now quite impressive data from a number of series showing efficacy of ABL class inhibition in ABL class PH-like ALL but much poorer response data for ruxolitinib, the type 1 JAK inhibitor in JAK-STAT-driven ALL. And so new therapeutic combinations or approaches are needed. Some of these include combinatorial treatment between JAK inhibitors and PI3K inhibitors, treatment with JAK inhibitors and BCL2 inhibitors, or directly targeting CRLF2 receptor itself, for example, with CRLF2 directed CAR T cell therapy. I would now like to shift gears a little and give another couple of examples as to how genomics are really revolutionizing our understanding of the classification of ALL. So this example that I'd like to discuss now pertains to ZNF384 rearranged leukemia. I showed this before on the Tisney plot of B-ALL, where we see the subtype of leukemia with distinct gene expression profile, the majority of cases of which have rearrangement of ZNF384 as shown. But we also see these rearrangements in another form of leukemia, that is B-myeloid mixed phenotype acute leukemia. And you would be aware of this phenomenon of mixed phenotype acute leukemia, where leukemias do not fit neatly into a single lineage of myeloid, T cell, or B cell, but typically express myeloid markers such as MPO, myeloperoxidase, and a lymphoid marker such as CD19 for the B cell lineage or CD3 for the T cell lineage. These tumors can either be truly biphenotypic, where we see a single population of blasts expressing multiple lineage markers, or we can see these bi- or trilineal leukemias where we see multiple populations of blasts in the same case expressing different markers. We became interested in studying mixed phenotype acute leukemia some years ago, where we were examining the genetic basis of early T cell precursor leukemia. Now, mixed phenotype leukemia has similarity to ETP-ALL, but some differences in the nuances of immunophenotypic criteria that are used to diagnose the disease. So we had embarked on a large-scale genomic analysis of childhood mixed phenotype acute leukemia. There are several other studies that have done this in both childhood and adult mixed phenotype acute leukemia, but the key parts of the data and the study that I wanted to show today pertain to the B-myeloid mixed phenotype acute leukemia cases that express CD19 and myeloperoxidase. And here, half to two-thirds of these cases have exactly the same fusions of ZNF384 as I was showing before for that subset of B-ALL cases. They have the same fusion partners that are rearranged to ZNF384 at the C-terminus of the fusion oncoprotein. The fusion partners are interesting. They typically involve chromatin remodeling genes or transcriptional activator genes. But we see no difference in the genomic features of the B-ALL cases or the B-myeloid mixed phenotype acute leukemia cases as shown here. So whether we look at co-lesions between B-ALL and B-myeloid cases, these are very similar. And if we look at the gene expression profiling data, again showing a Tisney plot for a number of representative cases of leukemia, we show that the ZNF384 rearranged cases have a very similar gene expression profile, irrespective of whether they're mixed phenotype or conventional B-ALL. So here this strongly suggests that this distinction between B-ALL and B-myeloid mixed phenotype leukemia in cases with this fusion is artificial, and really the fusion should be used to identify and classify these cases rather than starting with immunophenotype. It also raises questions as to why do we see this immunophenotypic plasticity, and why do we see these multiple subpopulations of leukemic cells within the same case? And so we performed several approaches to try and address this. The first was a genomic one where, for example, we took a number of cases and flow sorted subpopulations and sequenced them to see if there were differences in their genetic makeup, and really there weren't, that basically the lesions were the same regardless of the immunophenotype of a subpopulation within a single case. A second related approach to this was to again flow sort leukemic cells and then transplant them into immunocompromised mice and see what sort of tumor engrafted in each of the transplanted recipients. And what we found was that in a number of tumors, here again a ZNF384 rearranged tumor, it didn't matter what tumor population was transplanted, the resulting tumors reconstituted the diversity of the original diagnosed leukemia. So this strongly suggests that the plasticity seen in these leukemias is intrinsic to the cell and intrinsic to the patient. There are several other lines of evidence that support this. There's also supportive data from looking at diagnosis relapse data. We've published this in this paper and also recently in a large-scale study of diagnosis relapse leukemia, where we often see that the leukemia lineage can change over time. At diagnosis, it might be mixed phenotype. At relapse, it may be B-ALL or vice versa, but the founding lesion, such as one of these fusions, is exactly the same. It strongly suggests that the right founding lesion in the right progenitor confers this lineage plasticity. And further evidence in support of this is shown here, where we took a number of different subpopulations of hematopoietic stem and progenitor cells from diagnosis samples from these patients, flow sorted these progenitors, and then sequenced them for the founding fusion and secondary mutations, and we could find that a subset of hematopoietic stem cells, as well as the subsequent progenitor populations, but not non-leukemic populations, did have these founding fusions. So this strongly suggests that these founding lesions are acquired in a subset of hematopoietic stem cells, and that confers the lineage plasticity that we see in these cases. Another way of showing this is shown here. This is, if you like, a revised view of the hematopoietic hierarchy, where normal hematopoietic cells retain bilineage potential until quite late in development, and this in part explains why we see most commonly T myeloid or B myeloid tumours in mixed phenotype acute leukemia. This also has therapeutic implications, and an example of this is shown here, where if we look at the gene expression profiling of these ZNF34 rearranged leukemias, they have very high level expression of FLT3 through non-mutational mechanisms. FLT3 is typically not mutated in these leukemias. If we look at gene expression right across the landscape of ALL, ZNF34 rearranged leukemias have the highest expression of FLT3, even higher than MLL rearranged leukemia, as shown here. And some serendipity has shown that patients may respond to FLT3 inhibition. This is a case of a patient treated by Tim Lay at WashU. It was actually someone in his laboratory at the time who had multiply relapsed leukemia, and he sequenced the genome and transcriptome and found that this individual did harbour a ZNF34 rearrangement in their leukemic cells and also had extremely high level expression of FLT3, and treated the patient with sunitinib, resulting in rapid clearance of leukemia and the ability to proceed to a bone marrow transplant, and he remains in remission today. So again, another example where a somewhat unexpected vulnerability has been identified from genomic analysis in this type of leukemia. Just to conclude the talk, I want to shift gears really away from what I've been talking about so far, and that's talking about the different leukemia classifying events, to start to think about secondary genomic lesions and mechanisms of genomic evolution. We spent a lot of time talking about somatic lesions that are leukemia initiating and how they can drive cell intrinsic resistance, but we now understand that there are secondary and evolving somatic lesions, as well as germline lesions, each of which can influence leukemogenesis and drug response. And also this drug responsiveness can be related to perturbations in which leukemia cells interact with the bone marrow microenvironments, with the bone marrow niche. And I'll conclude by showing one example of these data now. And so this is a story that started many years ago that we first embarked upon when we asked the question, why do we see one fusion oncoprotein BCR-ABL driving two different diseases, that is acute lymphoblastic leukemia and CML? And one of the main findings of this study was that alteration of the Ikaros or IKZF1 transcription factor gene was a hallmark of ALL or CML when it transitions to lymphoid blast crisis. So these two lesions drove lymphoid lineage. A note about the genetics of Ikaros. So we now know that Ikaros alterations are a very important hallmark of high-risk B-ALL. Apart from the leukemia initiating lesions that I've shown you so far, Ikaros alterations are really the strongest and most powerful predictor of outcome and relapse in ALL. So Ikaros is a transcriptional regulator. It remodels chromatin. It's required for the development of all lymphoid lineages. And the main types of alteration as somatic events in Ikaros are shown here, We commonly see deletions, particularly a focal deletion that removes the N-terminal DNA binding domain of Ikaros. And we also see a number of point mutations as somatic events, typically clustered in the DNA binding domain here, or in the dimerizing domain as shown here. And in part, these lesions act as dominant negatives. They can relocalize Ikaros away from its normal site of binding at heterochromatin and redistribute Ikaros around the nucleus or indeed around the cytoplasm as shown here in these immunofluorescence images. We and others have done a lot of work understanding some of the mechanisms by which Ikaros drives a high-risk leukemia in pH positive and other types of leukemia, as well as poor response. In mouse models of leukemia, we've shown that Ikaros alterations synergize with BCR-ABL to turn a CML to a BALL, and also that these leukemias are quite resistant to chemotherapy, be it conventional chemotherapy or TKI therapy here with DNIP. And much of this resistance is due to aberrant cell adhesion, where Ikaros mutant cells adhere to each other and to stroma and extravasate into the bone marrow niche. And this is microscopy of cell cultures showing these Ikaros mutant cells adhering to each other. This is because when Ikaros is mutated, it de-represses expression of adhesion molecules and stem cell pathways. So we see de-repression of adhesion molecules such as Thi1, integrins, and selectins. And we see enhanced self-renewal. This is shown here with enhanced colony-forming ability of Ikaros mutant cells and acquisition of stem-like gene expression programs as shown here. This story has now become even more interesting because we now recognize a syndrome of germline predisposition to leukemia defined by germline Ikaros alterations. So this story began with this family that was identified with a child that was found to have a truncating mutation in the germline of Ikaros in the DNA binding domain as shown here. The same mutation was identified in his uncle, who also had leukemia, and a number of relatives, so his mother, siblings, and a niece, who all carried this Ikaros mutation. And similar, but not actually identical, but other germline alterations of Ikaros have also been found to define immunodeficiency and low B cell number, and indeed several of the relatives of proband also had features of immunodeficiency. So I use this as an example for a couple of reasons, one of which is to highlight the increasing importance of germline alterations in the pathogenesis of ALL. Often these discoveries have been made in certain contexts, but we now see germline mutations are important, both in familial leukemia, but also cases that appear to have no family history, sporadic leukemia, and also in specific subtypes of leukemia. Perhaps the most striking of which is p53 mutations in low hypodiploid ALL in children, but also the PAX5 G183 mutation in a subset of ALL, and germline ETV6 variants, which are probably the commonest germline mutation that are somewhat showing enrichment in hyperdiploid leukemia. So we see a similar pattern of both familial and sporadic alterations of Ikaros in BALL. This part of the study was led by Jun Yang at St. Jude, who'd sequenced several thousand cases of BALL, and after appropriate calling and filtering found that about 1% of BALL cases in children have a putative deleterious Ikaros variant. And there are additional lessons to be learned about the location of mutations and function in Ikaros, and I show a little bit of the data here. This has really been an important learning exercise. Previously, I showed you that somatic mutations are clustered here and here. The germline mutations are different. They're clustered in this region of the gene here, which is of less clearly defined function, as well as in the N-terminus. To understand the nature of these mutations and their functional effect, we used a wide variety of assays that look at simple transcriptional activation, cell localization, as well as looking at some of these more complex phenotypes, such as adhesion and drug sensitivity. Virtually all of them, with rare exception, showed abnormalities in one or more of these assays. 10 of these mutations or variants were subjected to a large battery of tests, and a couple of points can be made. The first is that if we looked simply at the transcriptional effects of Ikaros in vitro in standard cell reporter assays, we'd see that most mutations do nothing. And so these are data for two of the targets of Ikaros that show repression by wild-type Ikaros, de-repression by the somatic deletion mutant called IKZFI, but then virtually no effect, no de-repression from all the missense mutations, and the only deleterious variants were these ones that are either in the DNA binding domain directly or are truncating variants as shown here. However, if we look at these more sophisticated assays, we see that most of these variants are indeed very important. They cause mislocalization of Ikaros as shown here, as well as aberrant adhesion. Indeed, if we look at the relatives of the proband of the family, we see the same phenomenon. So these are immortalized B cells from the two siblings showing mislocalization of Ikaros due to the mutation. And the same thing is seen with adhesion. So again, I show in vitro data showing these clusters of cells. And indeed, if we do this systematically and look at every variant and then count the number of aggregates as well as score the size of the aggregates in vitro, these are empty vector control cells as shown here, but we see the vast majority of variants increase the number of aggregates or the size of aggregates or both. And indeed, these variants here that are the most deleterious on adhesion are in fact all germline variants. They are more deleterious than any of the somatic variants that we previously identified. So again, why is this important clinically? Well, obviously it's very important for understanding biology and disease predisposition, but we also think it's important therapeutically or potentially important therapeutically. This is a transplant assay where we can generate leukemia of various Ikaros genotypes and look at adhesion in vivo. And so here we're seeing leukemic cells marked red. These are the somatic IKZFI mutant cells. These are germline mutant cells showing this abnormal cellular morphology and mislocalization in the bone marrow. These cells become very resistant to therapy. This is ex vivo cell killing data where we find that most of the germline mutations become profoundly resistant to dexamethasone and dasatinib as shown here. So again, more resistant to therapy than we see with the index somatic mutation IK6. And this can be therapeutically addressed because in many ways, in fact, but one striking way of reversing this drug resistance is through FAK inhibition as shown here. This is important because as these cells adhere to each other, they activate signaling through integrins that are mediating adherence, and that activates the focal adhesion kinase pathway. And we see if we co-treat leukemias that are Ikaros mutant with dasatinib plus a FAK inhibitor, we can markedly prolong survival of inoculated mice, and we can completely reverse this aberrant adhesive phenotype in vivo. So these are very encouraging data that there may be ways to further improve TKR responsiveness in Ikaros mutant leukemia. So just to conclude, how are we using sequencing in leukemia management? So I show one example of this from Total Therapy 17, the frontline study of ALL at St. Jude. All patients enrolled on this study are sequenced at diagnosis with both transcriptome sequencing and whole genome sequencing. And then this information is used for a variety of targeted therapy approaches and risk adaptation approaches. This information is very important, not just for using targeted approaches such as kinase inhibitors, but thinking about stratification and other therapeutic approaches. And so here there are a number of implications of these new subtypes of leukemia, including modulation of treatment intensity, potentially reducing intensity for good risk subtypes, and then we can see a number of potential new therapeutic approaches that are being tested or have been shown to be efficacious already in preclinical models, several of which that I've mentioned throughout my talk today. I just finish on the point that although genome sequencing is obviously a complex proposition and is not simple either technically or from the analysis viewpoint, transcriptome sequencing is very powerful, and I made this point at the start of the talk. And indeed, while not truly comprehensive in giving us all information about the leukemia genome, transcriptome sequencing provides most of the information we need for risk stratification and tailoring of therapy. Examples of this are shown here. So here is now a clinical grade t-SNE plot where we've taken two cases here and classified them according to their transcriptional landscape. This is a case with ETV6 RUNX1 leukemia that was originally missed because of low-level fusion expression, but found to cluster with the ETV6 RUNX1 group, and the fusion was then rescued on manual inspection and curation. Another striking example of a case with no MLL rearrangement, but clustering with the MLL group, and was found to have an alternate alteration deregulating HOX gene expression, and so a case of MLL-like leukemia. And an additional point that RNA sequencing itself can be used as a substitute for some aspects of karyotyping, including aneuploidy assignment. And an example of this is shown here, where the integration of gene expression levels and variant frequency across each chromosome can be used quite accurately to assign ploidy status of individual chromosomes, and indeed can actually be found to correct misassignments of aneuploidy that can sometimes be an artifact of conventional karyotyping. An example which is shown here, a case reported as trisomy 15 that actually had a gain of 14, 2 copies of 14 rather than a gain of 15, as shown here on transcriptome sequencing. So there I conclude that genomic sequencing has revised the taxonomy of ALL. Transcriptome profiling is a key integrative marker of ALL subtype. The underlying genetic drivers of each subtype are highly heterogeneous in type and spectrum, and secondary genetic alterations are important determinants of drug resistance and can generate often unexpected therapeutic vulnerabilities, the example of which I've shown today being Ikaros. There is an increasing appreciation of the importance of the germline genome in driving leukemogenesis and drug responsiveness, and I've tried to show throughout multiple examples of where sequencing-based approaches really are important for accurate diagnosis, risk stratification, and tailoring of therapy of ALL. I conclude by thanking members of my lab, many collaborators who have contributed to work that I've shown today, both at St. Jude and around the country, and my multiple funding sources that have supported this work. And I thank you for your attention.